Right. So good afternoon, everyone, and welcome to this first webinar of the webinar series on research and winemaking. So today we will discuss about winery cleaning and sanitizing. So I'm Dr. Aude Watrelot. I'm an assistant professor and extension specialist of enology in the Department of Food Science and Human Nutrition at Iowa State University. I'm co-hosting and co-organizing this webinar and these webinar series with Mr. Drew Horton, who is from the University of Minnesota. He is a research specialist of enology, and he will be managing questions about the winemaking aspect. So before we start, I wanted to provide, some instruction about the webinar. So it's being recorded and that may be available by request. So this webinar would be two presentations followed by Q&A section, about 10-15 minutes. So for your question that you may have during all the webinar, please type your question either in the Q&A box or in the chat box. So on the bottom of your screen, you should see 3 dots. If you don't have the Q&A box on your right side of the screen, you should click on the 3 dots and the Q&A box will appear. And so, as I said, we will answer your question at the end of the webinar. Also, at the end of this webinar, you will receive an email with a survey, a link for a Qualtrics survey to complete. So we are always looking for feedback just to know what we can improve, what we can do to fit your expectation. So please take just 2 to 5 minutes to complete the survey at the end. And so today, that's a great pleasure to have Mr. Luke Holcombe and Mr. Cory Marx from Scott Laboratories and UC Davis with us, who will present the theoretical aspect and practical aspect of winery cleaning and sanitizing. And so thank you very much, Luke and Cory, for accepting our invitation to this webinar. And so now I will just leave Drew introducing Luke.
Yeah, thank you, Aude, very much. And thank you to all of our attendees. This is our first webinar and what we hope to be a series, an ongoing series. Without much further ado, I'll go ahead and introduce Luke. I've known Luke for 6 or 7 years now, and Luke is a very helpful guy for either a winemaker or an enologist to know. Luke was born and raised in California's Central Valley, was surrounded by a lot of wine grapes, and I assume a lot of raisins too. This piqued his interest to pursue a degree in enology and viticulture at Cal Poly in San Luis Obispo, a wonderful new program there. Prior to joining Scott Labs, Luke was assistant winemaker at McManus Family Vineyards in Ripon, California, and for a couple of years was the head winemaker for Post Winery in Altus, Arkansas. He currently resides in Lubbock, Texas with his dog, Mr. Wilson. Take it away, Luke. Thank you.
Awesome. Thank you. Only change there is I no longer live in Lubbock. That was a short, brief interlude there. Actually live in Bentonville, Arkansas, the home of Walmart. So thank you guys for the opportunity. Really enjoy doing webinars and presentations and things like that. And the world being the way it is now, webinars are the way. So thank you very much. And hopefully we don't have any technical difficulties. If we do, we'll just adapt, improvise, and overcome. So Drew asked me to do this, and one of my favorite things to do is give practical, applicable information that people can take right from the webinar and go start doing it and try to make as much sense as possible. So I've been doing that. My primary role at Scott Labs is sales rep, but due to my background, I do a lot of this sort of thing with webinars and education and presentations. So A little bit about Scott Labs. We have been one of the leaders in the winemaking, cider, and other industries for nearly 80 years. We were one of the pivotal people around maintaining yeast strains through Prohibition. Basically invented active dry yeast, among some other things. A lot of people know us for our yeast, which is awesome, and our enological materials. But we also have equipment from the crush pad to pumps to filters, cross flow filters. We also do, we're the sole supplier of Velcro to the industry. And we also have a very large cork and packaging department as well. So that's a little bit about us. If you'd like to know more, contact your local rep or call into the office. So a little bit about me. There's my ugly mug. So that's my name. There's my Instagram handle. I have a bachelor's in enology from Cal Poly San Luis Obispo. One of the lucky things I was able to do was actually apprentice and study under Dr. Bruce Saucklin, who, if you're not familiar with Dr. Bruce Saucklin, he wrote the Bible, essentially, of winemaking. I was at Virginia Tech for many years and took a sabbatical out to Cal Poly. So I have about 15 years of commercial large-scale wine production. I've done everything from hand bucketing, 11 tons of fruit off the back of a truck and making wine at the micro scale, all the way up to 75,000 gallon fermentations and kind of everything in between. So I've done bulk wine receiving, bulk wine shipping. I've done screw caps, corks, bag in the box, large bag in the box when you fill a container and ship it overseas, so on and so forth. So I've really been exposed to quite a bit of stuff in my tenure, and it's really helped me along the way. So about 20 million gallons. I kind of lose track, somewhere along 10, 15, 20 million. I'm not sure where I'm at. So it's a little bit about me. So, I like to try to give a perspective of like what we're trying to do and kind of give an overview. So I want to break these things down. Overall, this is hygiene, right, in the winery and in the wine cellar. And it's a combination of three particular specific things, which is cleaning, sanitizing, and sterilization. And I'll define those here in a second. Hygiene is a means of microbial control, product integrity, equipment maintenance, environmental management, and conservation. So that we're trying to do all those things at one time. These are inexorably linked. So without proper microbial control, you lose control of your product integrity. If you're not maintaining your equipment, you will probably have a microbial problem, which will then affect product integrity, and on and on and on it goes. So also, hygiene can be fun, and shout out goes to my friend Fabian over at Noisy Water in New Mexico for this little gem here. So this is, we'll talk about this a little bit more in a second, but here he goes. One more time. So basically what's going on there in preparation for harvest, I don't know if you guys saw that, but hopefully it worked. He's trying to launch the hose ball that was cleaning the hoses as far as he could. It's kind of a joke on seeing how fast they could do it. So you can have some fun with it. Set up maybe a little cellar Olympics and see how far you can launch the hose ball. So kind of a fun deal. So from a legal perspective, the producer, you guys do have a legal obligation to produce wine that is pure, wholesome, and free from adulteration and contamination. It's also a legal responsibility for operating the winery in a sanitary manner according to regulatory agencies. Now, historically in the past, we've really only been under the auspices of the TTB, and so long as your gallonages and taxes were up to date and you were reporting correctly, that was about all we had to be concerned with from a regulatory agency standpoint. However, that is no longer the case, and it will be changing and evolving, and the regulations and the oversight will continue to grow and get a little bit more onerous. So you're going to have probably county health commissioners that are going to have a play, state ABC boards that are going to have a play, and then at the federal level, not only are you going to have the TTB, but you're also going to have the FDA. So if you want to know more about that, you can Google FSMA, which is the Food Safety Modernization Act, I think of 2013 is what it was, but really it started classifying wineries and cideries and things like that as food production facilities and therefore regulated as such. So There are some spotty spot checks going on right now that the FDA is conducting. It's not as well as it's going to be, but it's something to be aware of and really start developing a plan before it actually happens. So that's all I'll say about that. So these are my definitions for the 3 different stages of hygiene. So we're talking cleaning, which is a process used to reduce or eliminate debris from a surface, right? So this is like washing your hands, right? And you would do this on the inside and outside of wine presses, hoses on your floor, your drains, et cetera, et cetera. Tools that we'd use for this are brushes, rags, liquid circulation, hose balls, which is what I'm showing there, and that's what that video was showing, was pushing that out of a hose. And the first time you do that, if you've never done it before, you'll be shocked at what comes out. The water, especially if you're using something like caustic or percarb, it'll be black. And that is what this does is, it's one aspect just to run a recirculating chemical through, but a ball, for example, is actually physically scrubbing the inside of that hose and is removing soil from a physical action. So that's where we do physically clean. Chemically clean is going to be using, caustic or peroxide or something like that in order to render it chemically clean. And then also the rinse that's involved with that. You want to make sure that there's no residual chemicals there. Also microbiologically clean, and that kind of dovetails into sanitizing. So cleaning is just getting the dirt for the most part, right? Because once you get the dirt out of the hose or off of the press or off of the floor, then you can actually move to the next step, which is sanitizing. And this is the process of significantly reducing, and not 100% elimination, but significantly reducing the number of live microbes on a surface. So you do this with chemicals, potentially with heat, maybe UV light, things like that. By definition, sterilizing, on the other hand, is the removal or destruction of all the living microorganisms. And remember that sanitation is not sterilization. So when you say, oh yeah, I sterilized that, I ran KNBS citric water through that, that is not sterilization. Okay, so it's really important to understand these terms and use them appropriately. So from a validation standpoint, one of my favorite tools to use to know whether or not a surface is sterile and to validate my cleaning procedures, I really like to use ATP swabs. They're great. A little bit of an aside on that. These ATP swabs are used, and basically the sleeve that you put the swab in once you've swabbed your surface contains the luciferase angel, which comes from, luciferase enzyme, which comes from the angel of light from the Bible, Lucifer. And it's the same enzyme that's inside of the stomachs of, those fireflies, right? And so when you put that swab in there, the ATP, which is basically the energy, the fuel for life, it interacts with that luciferase enzyme and produces light. And then there's a meter that reads the amount of light produced off that. And the amount of light that's produced off that is relative to the ATP, which is relative to how many microbes are on a surface. It doesn't take the place of plating, for example, to know what sort of organisms you have. It's just a relative number of how much is there, okay? So cleaning is your first step. Sanitizing is your second step. Sterilizing is your third, and don't skip a step. Because if you try to go from, if you just gloss over cleaning and go straight to sanitizing and sterilizing, you will not achieve your goals. Real quick, here's a practical thing I think is really important. When you're talking scrub pads, when you're cleaning, it's really important, especially on stainless, to use white and not red or green. Red or green Brillo pads will actually scratch the surface microscopically of the stainless and create a breeding environment for microorganisms to hang out. Now, one could say that if you had a dedicated tank for cold stability, this might be a great tank to scratch up with a green or red Brillo pad because you create more nucleation zones for the tartrates. I wouldn't recommend that, but if you have used a lot of those scrub pads, you'll probably notice that there's a lot of crystals that develop on that wall compared to other ones. So it's just a real quick practical thing. So efficiency is important, especially on the smaller scale because, and larger scale because of more monetary reasons, but on the smaller scale because for every action, whether you're moving 5 gallons or 50,000, you kind of have the same basic setup parameters, right? The same amount of energy input insofar as somebody's got to drag that hose and hook it up to the pump and then start running the water and so on and so forth. So that cleaning step, the cost and the labor associated with it when you're at pumping 50,000 gallons is diluted much more than if you're making 500 gallons, for example. So we want to try to be as efficient as possible just to make everyone's life easier. So when it comes to cleaning, you want to watch your SSOPs, your standard operating procedures, and it's the watch rule. So this is water, action, time, concentration, and heat. And you want to balance these for optimized efficiencies. A parlay would be picking the right kind of yeast, and one yeast is not going to do every job in the world. So you kind of have to look at your grapes and what sort of microbial competition there may be, what sort of alcohols, their pH, so on and so forth to pick the right yeast. Same thing goes in cleaning. You have to look at a number of variables. So water, super important, right? Water is king. Water is life, and it has a big impact on how efficient you can be during cleaning and how it interacts with not only the surfaces but also the cleaning chemicals. It can contain impurities, so wine industry is pretty lucky when it comes to alcoholic beverage production because of our low pH, our usage of potentially SO2 and other products like that, our adoption of filtration. We're a relatively microbially stable beverage, whereas with beer, for example, they have to be much, much more careful because they have lower alcohol. The pH is so high, and they can't use SO2, so on and so forth. They want to package things hazy, on and on. So they're much more concerned with their water because beer is like 95% water. So they do a great book to pick up is The Practical Brewer. It goes into water and does a lot of in-depth analysis on what you should be looking at. But when you look at water, it's 100% of your rinsing solution. It's probably about 97% of your cleaning and your sanitizing solution. So it actually has a huge role. A good takeaway generally is that you need about 10% by volume of water for the vessel being cleaned. Okay, so for a 55-gallon barrel, 5.5 gallons, on and on. 500-gallon tank, you probably want about 50 gallons in there. So now, can your processed water be reused? That's an important question. It can be, depending on what you're doing. For example, like a caustic wash, what you can do is when you're done with your caustic wash, you can actually use pH strips and measure that processed water. And if it's high enough, you could actually probably reuse it on the next tank. So this is where that efficiency can come into play. So mechanical action, that's the A. The key factor for success, especially in difficult reach areas. And what I'm talking about there specifically is entrapment zones. Entrapment zones are any place that are like, you can't really see, they're hard to get at physically. And so you have to rely on your cleaning solution and potentially something like a hose ball to get in there and actually have contact and conduct the actual cleaning, the scrubbing bubble effect, right? So you want to try to minimize entrapment zones. One of my pet peeves, and I've got a lot of them if you know me, but one of them is seeing hoses stored either in a circle, on a loop like you would a water hose, or draped over a number of tanks and seeing all of these dips in the hoses. Well, anywhere where you have those dips and those low points, there's inevitably stuff is going to pool there and be a potential contamination source. So you really want to try to let that hose drain as much as possible and set it up for success. But you can also see this in piping on like bottling lines, fixed installations where whoever made it really didn't have an understanding of sanitary execution, and there's a lot of dead zones. And so you really have to take that into consideration when you're looking at a cleaning system. So the action forces the contaminants off the surface, and it's used in conjunction with dissolving, right? Because the solution to pollution is dilution. So part of what a water-based cleaning solution does is it actually allows for the soil and the dirt to dissolve into the water, right? And these reactions are often equilibrium controlled. Constant circulation is required. So the old bachelor soak maneuver, where you just put all your dishes in the in the sink and throw some soapy hot water at it and just let it sit. Works partly, but most of the time we're talking circulation and even impingement. And also I'll kind of explain impingement in a later slide here. So you got to keep good circulation going. So here's just a real quick guide on what sort of flow rates you need for the different types of soils. You know, you'd be surprised sometimes how much water is required to adequately wash a tank, and sometimes that's not just 10%. It may be dictated by the speed of the pump and how much surge volume inside of the tank you need to feed that pump at the speeds that you're running it. So sometimes it's not just at 10%. You may start at 10% and go up from there depending on the speed of your pump. For hoses and piping, here's a guide on 5 to 7 feet per second. So you need about 24 gallons per minute, 1.5 inches, 69 to 96 for 2.5 inch hoses, and 2 inch would be somewhere in the middle. So you want that turbulent flow, right? So if you're pumping really slow through a piping or a water system, what'll happen, or a hose system, what'll happen is you'll get the bottom, or maybe the bottom half of the hose, but you actually won't get contact with the top of the hose. That's where the hose balls are so important. That's also where you could create some back pressure on the back end of the hose in order to make sure everything's filled up. So it's really important to look at that stuff. So contact time. Cleaners do not work instantly, so you can't just like spray caustic on something and say, "Yep, and we're good." Sometimes it needs actual constant time to start breaking down those proteins and some of the biofilms in order to loosen it. And then there are also some other cleaners that are built solutions, which I'll talk about here in a minute, that actually chemically like lift the soil off of the surface. And this best way to explain this is like, if you just try to wash your hands with water, nothing will happen. But if you put soap on it, it actually helps lift the dirt off of your hand and allows you to actually get down to the surface of your skin where you can clean it. So consider how a cleaner is being applied insofar as contact. So if you atomize a spray, for example, like something out of like a backpack sprayer or a weed sprayer, if you have really small droplets, you maybe get better coverage as opposed to a big solid stream. So you kind of have to, that's where that concept of impingement comes in again, and what you're trying to do. So sometimes you can spray stuff down, you can soak things insofar as like a COP system where you're putting fittings and gaskets and things like that actually in a container and filling that container with some sort of cleaning chemical. There's also foams and gels, and certain cleaning chemicals actually have a foaming action to them. And this helps for those entrapment zones that we were talking about earlier. So,
Luke, I'm sorry, I'm going to interrupt just for a second.
Sure.
With a friendly 5-minute warning.
Okay, I'm going to go faster.
Thank you.
So this is a real quick look at how much soil is adhered to the surface based over cleaning time. And basically what they're saying is if you do nothing else, at least rinse with hot water, you'll get a lot of stuff just right off the surface. So the concentration, again, this is the C part of watch. The amount of is used is dependent upon water quality, quantity, your soil quantity, and quality. Is it damp? Is it dried? Is it baked on? And the temperature of the cleaning water. And we're not talking about this kind of concentration. We're talking like parts per million, pounds per thousand, things like that. So don't think real hard about it. The little works, a lot is not necessarily better. Heat. So every detergent and cleaning chemical is really dependent on temperature as well. Some of these things like caustics are actually exothermic reactions where they will produce heat. And the load carrying capacity of water in order to absorb dirt and soil is actually based on temperature, and the warmer it is, the more it can. So it has to do with how the molecules are spread apart, but won't focus on that too much. So considerations for cleaner selection and concentration. Again, take a look at what surface you're dealing with. Like this is a really dirty tank, obviously a lot of tartrates, red wine color. There's a lot of things there, and that's going to take a really strong, probably a caustic solution to start with, and some sort of impingement cleaning to blast that off there. You can soak it with hot water for a long time, but an impingement with a chemical aid is going to be the best way to do it. So alkalis and acids, I'm kind of going to blow through this. You can Google a lot of this info, and because of that, I think just put it up there. Sometimes you need to passivate stainless steel. So we kind of do this unconsciously in the wine industry because of our low pH, and we use citric acid all the time for cleaning, so this helps. But in the brewery world, this has to be done on a regular basis. And if you want some more information about passivation, there's a link there. But here's a general, like, caustics are above a pH of 13, the acidic pH is 1 to 6, on and on. So that's kind of basic stuff. Real quick, caustics, potassium or sodium hydroxide, why would you use one or another? Sodium is cheaper. However, if you are pumping into a wastewater system, I would suggest potassium, because potassium is actually a nutrient for microorganisms, whereas sodium is a salt, and it's kind of won't help your septic system. Percarbonates must be coated. Go into too much of that. So they're very highly corrosive and reactive. It increases surface tension. Hard water and heat will lead to scale. It can denature and char soil and potentially redeposit it, and that's what this looks like. This is a caustic film, and if you have this red hue on your stainless, it's time to get something like this D-Stainx and get in there and take that off, because that could actually be harboring microorganisms. So how much cleaner do you need? Here's a guide. If you want this guide, I can send it to you. It's based on our D-Stainx products. It's kind of like, hey, maybe I should use a little bit more if it's really heavy. Water temperature maybe change if there's heavy soil, and so on and so forth, right? So there are dose rates out there. And again, one of my best tricks up my sleeve is we could call me with the question. I Google it, and I can usually come up with the answer if I don't know it. Choosing sanitizers. Real quick, you have iodines, quats, ozone. I think iodine and quaternary compounds are becoming less popular these days, so I won't spend much time on them. Ozone's great. It's an oxidizer, which has basically the same effect that like bleach would have. It's an oxidizer. Percarbonate, hydrogen peroxide, all these things are oxidizers. They're broad spectrum. And ozone, what's nice about it, it's really, really dangerous in its active form, but it breaks down pretty quickly into oxygen and water. So it's pretty safe once it's gone through its half-life. Chlorine dioxide, actually, this is great for water sanitation. I really like it for that. It's pretty stable. It can be expensive to generate, so it's kind of in the same area as an ozone generator. And there's options for that if you're interested in it. I can point you some resources. Peroxy compounds, so this is sodium percarbonate, peroxyhydrate, or something like our D-Stainx. Our D-Stainx is percarb plus some conditioners and things like that. SO2, inexpensive, fairly effective, but it is pretty rough to work with. So, oxidizers, chlorine, bleach, really common in the household, but they can lead to formation of TCA. So no, don't use them. Do not use them in the winery. I actually have customers who are battling with environmental TCA right now, and it's a very, very difficult thing to eradicate. And historically, it's been, well, time to bulldoze the facility and start over. It's that bad. So, and this is cork taint, quote unquote, right? TCA, trichloroanisole. It's corrosive also, right? So you can really rust some things, including stainless. It's a really strong oxidizer. That's why we use it. This is what a clean tank looks like. It's kind of a low-res picture, so sorry about that. What you have right there, this is, I believe, a Leckler tank washer right there. There's Leckler, there's Cloud, there's GammaJet. They all have their pros and cons. But essentially what it does, you hook this up to a pump, a high-speed pump, usually a centrifugal, and you circulate this cleaning solution through that. And what it does is it rotates on 2 axes, and given the size of the tank and how fast you're pumping, it'll eventually get 360 degrees all the way around on every axis, and it'll blast the tank with a properly impinged water stream. So those are really great. They come on carts. They work really well. They're kind of complex machinery inside, but if you don't have one of those, they're usually about $5,000, maybe a little less depending on the size. But a certain really large brewery has one of those installed in every single tank. And if you want to do professional tank cleaning, this is how it's done. Let's see here. So real quick, last couple slides, pre-harvest prep. One of the things I like to do is with hoses, I actually like to cut them before harvest right about there and reinsert the ferrules and reband them, because if you can notice where that hose is bent, that's a primary stress point right there. It's always getting bent right there, and that's a common fail point. So not only do you prevent a hose from blowing up, but you also are able to clean the hose in a specific area of the hose that's probably the worst offender for contamination, which is in between the hose and the ferrule. So cut it off, sanitize it properly, reassemble. Pump maintenance. You should be doing this pretty much before and after every single time you use the pump, but definitely at least before and after harvest. Take that puppy apart, put new seals in it, clean it properly, and make sure it's clean. This is, I think there's a certain pump out there which will remain nameless, but it's a pretty popular one that I think is the primary source for spoilage in the wine industry. Want to know more, contact me later. Drains and floors, clean and sanitize the floor often and before harvest. Take your drains apart, scrub them, use a foaming cleaner of some sort, because I can walk in, and Drew, I'm sure you've seen it before, you can walk in a cellar and know if they've got a microbial problem or not. You can smell it almost instantly. And it's really refreshing when you go into a clean winery because you're like, wow, it smells amazing in here. So that was a lot of information very quickly. I apologize. I talk fast, so hopefully all that's there. And maybe if it's available online afterwards, you can slow me down to quarter speed and actually hear everything I'm saying. So there's all contact info for Scott Labs, and I'm available to be reached there as well. So that's it.
Good. Thank you very much, Luke, for your great presentation. That was a good overview of everything that you should be doing in a winery before harvest and during harvest. So now that's my pleasure to introduce the next presenter. So Cory Marx, who is a PhD student from UC Davis. So he is in the third year PhD in the Oberholster Lab Group at the University of California, Davis. Cory's research focuses on applied cleaning and sanitation of winery fermenters and cooperage. He spends harvest outside the lab working production in wineries in the Napa Valley. So Cory, if you want to start presenting.
Great, yeah, here we go.
Yep. Thank you. And
awesome.
Just a reminder, please type your question for Luke or for both of the presenters in the chat box or the Q&A box, please.
Great, thank you, Aude, and thank you all for tuning in. I think this is perfect timing for a talk about cleaning and sanitizing, probably as most of you are starting to go through veraison, and actually as we start to bring grapes in here in California. So this is going to be a really good complement to Luke's presentation, not only because I'm going to be singing the same song in a lot of ways, but also because I'll have some data to show from my own research that will actually put some, quantitative evidence to why this stuff is really, really important. And the way I want you to look at this presentation is really about thinking about how you intelligently develop and monitor cleaning and sanitizing protocols for success, not only in current fermentations and what's about to go in the tank, but also down the road. While it seems like all you do during harvest is clean and sanitize, wine's the long game, not only in terms of élevage and bottle aging, but also in terms of, years down the road. And Luke suggested it there when he talked about biofilms, especially in places that are in these shadow areas where you can't get good mechanical action. If you don't do the work now to prevent those robust biofilms from forming, you're going to have endemic sources of contamination year after year. And frankly, a lot of those parts are nearly impossible to clean if you have dead space or bends in hoses, pipes, things like that. And the real focus of this presentation will also be, and I'll sound like a broken record, that established written protocols ensure the product quality and worker safety. Even if you're the only person in your cellar, when you're running around in the hustle and bustle of harvest, it's super easy to forget a step. And that's how you have problems down the road. So as Luke also talked about, part of the difficulty in the winery is there's a wide diversity of not only materials, but also designs, configurations. No two wineries are the same. While I think when most people think about cleaning and sanitizing, they often think about their tanks. I would argue that that's probably the easiest part to clean and sanitize, And studies have actually shown, which I'll talk about later, that this part, the tank and the bottling line, tend to actually be the cleanest parts of the winery, just because it's like, okay, wine's about to go in here. I got to make sure this part's clean. But it's all your transfer systems and all the little bends and cracks in your tubing that really go about spreading contamination in the winery. And again, we're going to separate cleaning from sanitizing. Cleaning is all about removing soils, while sanitizing is about inactivating and killing microbes. And that'll be really important, not only because they're different steps in the process that are generally separated or really are separated, but also because the products are marketed differently. So when you're dealing with your rep, you want to be specific in what sort of product you're looking for. So definitions, Luke did a really good job talking about the left-hand side of this. So I'm going to focus on the right-hand side of this slide and a little bit about safety. And like he has pet peeves, I've got pet peeves. And walking in the winery, when I see a poorly organized binder of all the SDSs, formerly called MSDSs, talking about all the chemicals, or the PPE is disorganized and not well labeled, that drives me crazy, because you really are risking the safeties of yourself and your employees by not having these things accessible and in good condition, frankly. Sometimes they're just these water-stained binders that are thrown together. But do yourself a favor before harvest kicks up too much and take a look through those binders. Make sure you understand these different symbols and what they mean. Make sure you've got the proper PPE. Make sure you also have the proper spill kits and mitigation strategies in case something goes wrong. So the framework that I've worked on for my research, and kind of a decent jumping off point for starting to form a cleaning and sanitizing protocol, is this 5-step process, which is essentially just a cleaning and sanitizing cycle that are separated by water rinses. And the purpose for this first water rinse is really just to remove the gross soil, the big debris. Not only is this going to make the cleaning cycle a bit easier so that you could, in theory, reuse your cleaning solution if you're going to go from tank to tank. But this will also, just take care of really the bulk of the load. And this could be hot water. Generally, we would caution against, certainly don't go all the way to steam, because you're going to end up cooking all that debris on and then causing yourself a bigger problem than you had to start with. But this will help lift some of the work away from that cleaning cycle. After you've done your cleaning cycle, that intermediate water rinse helps to remove the debris, because sanitizers tend to be even more susceptible to reacting with any remaining organics, and they'll totally lose their ability to effectively sanitize a surface if you've reacted whatever sort of chemical that you're about to use. This is also important because this isn't a steadfast rule, and there are many different types of cleaners and sanitizers, but often you have a cleaning solution that's alkaline and a sanitizing solution that's acidic. So separating them by a water rinse can help to make sure that you neutralize your system prior to adding, an opposing chemistry, so that you're not just neutralizing your sanitizer and then also losing its efficacy. That last water rinse, it's generally applied even when folks have chemicals that break down innocuously. I still see most winemakers doing a post-water rinse just because they're a little bit nervous. In the case of peracetic acid, people don't like the idea of having the breakdown acetate in the tank. So a water rinse can ensure that you don't have that. But again, I'm going to hammer home the point Luke said, not only now, but with some data later. You need to know the quality of that processed water that you're using, because I'll even show an instance in which I measured, using the ATP meters Luke talked about, some of the processed water in a local winery, and it was just ridiculously high with contamination. And if I hadn't done that prior to adding it to a tank, you basically would have undone all the work you did in the early part of that process. So cleaning, and these points, I'm going to, again, sound like a broken record, but I really think it's important to drill home what we're doing here. The proper cleaning regimen is the most important aspect. You simply cannot sanitize if you don't properly clean, for all the reasons Luke stated and for what my data is going to show later. There's no substitution for mechanical energy. As I stated earlier, the systems are different. When you have when you have hoses versus tanks versus your bottling line, you need to get those specialized tools to make sure that you're not doing one-size-fits-all cleaning and sanitizing, because that simply won't work. Dunking your, if you have a wine thief or a siphon or something and you just dunk it and don't get any mechanical action in those small tubes, you're really asking for a problem down the road, frankly. So there's a number of ways to do it, and they're all going to vary based on your system, your budget, the size of your winery, but those could be brushes, those could be spray balls, those could be atomizers. There's a number of different ways to do it. While contact times range from normally 10 to 20 minutes on the sort of extreme scale, they're going to vary based on the chemical and the concentration. Talk to people like Luke, talk to your supplier, use them as the resource, as the expert to help dial in what that contact time, what the temperature of your water should be. They're the experts. They're going to know how to do this wisely. Sanitizing, it's secondary to cleaning. It's your extra level of assurance. As I stated, they're really, really reactive, so they can lose their power. You need to make sure not only that you clean first, but that you rinse that cleaning solution well so you don't lose the efficacy of your sanitizer. As I just stated, the water rinse cycle is important, but really know the quality of your water rinse and have a way to understand if that quality varies based on the season, based on the time of day. I know, most people don't really think about just kind of using some sacrificial water when they start to use a hose, but the water that's been sitting in that hose overnight, if you're just starting to use it, is really pretty nasty. And I would urge you to at least just run it in the drain for a couple seconds. I know I worked in a winery where the water frankly stank when you started to use it, so you needed to take the time and actually just sacrifice some water before you could even make it be useful. No different than cleaners. Work with your manufacturers, work with the reps. They're the guys that can really help you out. So another note on TCA risk, I think Luke said just about everything you need to know. I'll explain the chemistry just a little bit. In addition to TCA, there's also TBA, which is the brominated equivalent. And the formation of these things has to do when you have phenolic material. So that's not only what's in wood of all sort, like corks, like pallets, like cardboard even, but also in grape material. And when these things are exposed to solutions like hypochlorite, you form compounds called halophenols or chlorophenols. When it's chlorine, it would be bromophenol if it was bromine. When you have those compounds and you have naturally occurring molds, they can actually transform these halophenols into the haloanisols. And the compound that's up there on the top right is 2,4,6-trichloroanisole. That is the cork taint compound. The sensitivity for these things, or the sensory threshold, is on the nanogram level. So when they're around, even in tiny, tiny quantities, you notice them and they're nearly impossible to sniff out. Hypochlorite or bleach is the main culprit for this. And not only does that happen with corks, but I don't want you to think that cork taint is limited to corks. This is also a problem in pallets, in cardboard, and it can come from other sources of wood in the winery. So I would strongly encourage you to remove those sources, or at least get them away from your production area as much as possible. Because I know sometimes when people bring pallets full of bottles near their bottling line, you can smell that musty kind of bagged baby carrot smell, if you want to call it that. Contrast hypochlorite to chlorine dioxide, which is a potent oxidizer. The way it works as an oxidizer is more similar to ozone than it is hypochlorite. It's more potent as an oxidizer, and importantly, it does not participate in that reaction to form halophenols, meaning that it is actually a viable sanitizer in modern wineries and can be a very good one for water. It breaks down quite nicely. If you think you might have a TCA risk or a TCA issue in wine, you can actually use saran wrap. It's a funny little trick, and sort of swirl it around in the wine for 30 seconds or so. Discard that saran wrap and then smell treated and untreated versions of the wine. The compound in saran wrap, or the coating, is actually works in sort of scavenging the TCA, and you can notice if you've got a risk that way. It's just an easy little trick. So not only is developing a protocol important, monitoring the success of that protocol is important. Luke introduced ATP bioluminescence swabs to you, and I'll actually show several slides with data on that. But I think that works really well in concert with the normal environmental swabs, which produce the kind of yeast cell colonies on Petri dishes that you see up there. And you can see a serial dilution series that I did. That's a monoculture of Saccharomyces. And if you burn that image into your mind, and I'll compare it to one of contamination that we see later, you'll see there's a big difference, really. There's other methods like PCR, other molecular methods. Those are generally restricted to in-house labs of like million-case wineries, really large facilities. But locally, you might have the resources to submit samples to one of these labs, and they'd be able to process them for you. So you could introduce that into your plan. So in an ideal world, you'd have all three of these things, but this isn't an ideal world. That's probably not realistic. I would highly encourage you, though, to consider adopting one of these methods, or potentially while you're developing your strategy for cleaning and sanitizing and choosing different chemicals, use some of these methods to actually monitor how successful you are. What I see almost 100% of the time is spoilage is only noticed once a batch of wine is spoilaged, and then you have to spoiled, and you resort to all these other tools to try to recover the batch as best as you can. They're more expensive, they're more time consuming. If you have a way before the grapes go in or before the juice goes in to actually put a number to check at the end of your cleaning and sanitizing whether or not you've been successful, you empower yourself as a winemaker, and you're really going to save yourself time and money in the long run. Now here's the fun part where I get to show some data and actually prove what we've been talking about. So these are ATP swab results. And what happened here is I had a number of different yeasts and then a mixture of different bacteria. And I formed biofilms on small pieces of 304 stainless steel, so that your normal tank material, by just soaking those for 14 days with a slight amount of shaking to simulate just normal fermentative turnover and turbulence. Those were then treated by, or first they were rinsed with water to produce a visually clean state. And then afterwards they were treated with cleaners and sanitizers. And what's really important to notice here is the difference between the left and the right. On the left side, these are your cleaners. On the right side, those are your sanitizers. So even though these were visually clean stainless steel pieces, there's a huge difference in the efficacy of the cleaners and the sanitizers. So if you were to just take what you thought was a clean piece of stainless steel and treat it with your sanitizer, you would have had a huge risk for contamination. You want to be on the left side. And here's a zoom in on the left side, and the green and red lines are what the manufacturers supply as recommendations. Below green would be considered a pass for a clean or a sanitary surface. In between is what they call risk for contamination, and above is contaminated. So even in the case of these cleaners, which had a huge reduction versus the sanitizers, they're not enough on their own. You can't skip the full process, the full cycle. Next, I want to show an experiment we did with much larger tanks, and we tried 10 different cleaning and sanitizing cycles. And you can see on the left side here the different sample locations. And on the right side, again, ATP, you sort of see a rainbow of colors here. What the different cleaning and sanitizing cycles are isn't important in this context. I want to show two different things. And the first thing I want to show is that big boom in purple on the left side with the circle around it. That's actually the winery process water, which is normally low, but you can see on this one instance, there was that massively high level of contamination in the process water. And I didn't even believe it. I had to double check with another sample, but if I had cleaned that tank with that water at the time, we would have totally contaminated it. The second thing that I want to show on this graph is those two generally large peaks that are large across most of the trials. Those are the valves. And the issue here is we have a top-mounted spray ball, and those are in the shadow. There was no part of the protocol that requires people to disassemble the valves and actually scrub them. So in this case, you would have had contamination no matter which protocol you tried. As we start to wrap up here, I'm going to repeat and talk about those major areas of microbial contamination. It's the winery tank surfaces and falling lines are generally clean. Really the drains and the area around the bungs of barrels that have high levels of contamination. And I did not only ATP swabbing, but also environmental swabbing of those tank surfaces. And here's one of the valves. You can see mold, you see a diversity of bacteria. It's really a nasty situation. Lastly, here's the strategy. Pay attention to those critical areas. adopt a strategy for monitoring and putting numbers on the success of your protocol, and have that protocol written with checklists. Have a smart selection of the tools you use. These brushes on the right are not only color-coded, so you know not to mix them with your cleaners and sanitizers, but this particular brand is also injection molded into the handle, meaning there's no gaps or no ability for those bristles to fall out. So many times you see people doing floors with the traditional broom type deals or scrub brushes that have the bristles that fall out. And then in those holes, you can start to develop colonies that you're actually just spreading as you move it around. 130 tip as sort of run out of time, I want to talk about cross-contamination with barrels. Barrels is really the focus of my research right now, so I had to say something about it. When you're going around topping off your barrels with a wine thief, be very, very careful, because there's no better way to spread contamination than dipping in one barrel and then going across to another and another and another, and along the line somewhere you have a contamination risk. Have a wine thief that can be heat safe, like glass, and carry around a little electric kettle and actually periodically turn that kettle on and just dip in between the barrels. It only takes about 10 seconds, but that way you can heat kill anything that would have been on that thief before you go into your next sample. If you also carry around a spray bottle of ethanol, just dose around the edge of the bung. You can see on this barrel how nasty it is from some sloppy transferring. That's my transferring, so I'm able to call it that. But we didn't have any issue. I always walked around with a bottle of ethanol to make sure that when I pull up the bung, you don't have anything fall in. And again, here's a recap here, but I want to leave time for some question and answer. So I'll just say thank you all. I think you'll all be getting a download of these slides. So my email's at the beginning. Feel free to reach out. Things get crazy during harvest, so I might not be super, super timely, but I'll do my very best, I promise. Thank you all very much.
Thank you very much, Cory. So remember to use the Q&A box and the chat box to write or to type your questions.
And while you're taking a minute to write your questions, I just want to state I'm obliged to make a little statement that some of the material presented today is based upon work that is supported by the National Institute of Food and Agriculture and the U.S. Department of Agriculture under award number 2018388640-28146 through the North Central Region Sustainable Agricultural Research and Education Program under project number ONC19053. The U.S. Department of Agriculture is an equal opportunity employer and service provider. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the authors and do not necessarily reflect the view of the U.S. Department of Agriculture. Thank you all. I'm going to go ahead. We have one question on the Q&A that I'm going to read, and then I have a couple of follow-up questions myself, and we still have a few minutes. Is that okay, Aude?
Yes, I think we have like 5 minutes or maybe a little bit less, but we may go a little bit over the time. That's
okay.
Good. Thank you.
So here's a question I get often, and we'll keep the reply brief. I think I already know the reply. The question is for a small-scale winery where they have stainless steel tanks below 8,000 liters, so 2,000 gallons or less. Can we clean and sanitize with steam? And they give the example if they rinse immediately with water straight after they use a tank, and then just use steam, how effective will that be? Either one of you can answer, but you have to.
Short answer, no.
Short answer, no. Cory, do you have any comments about that?
Yeah, I would hesitate to recommend that, only because, especially in the case if you're using a caustic or an oxidizing agent, you're actually physically breaking down things like biofilms. Biofilms can be hydrophobic, meaning they're actually water repelling. And I know I've seen surfaces where you can actually tell there's a film there because the water beads up sort of strangely on it. There would be no amount of just putting water on it that would solve that problem, and actually going to steam or too hot of water could really compound that problem and sort of cook that biofilm onto the surface. So I would hesitate to recommend that, although again, have some way of validating. And I think what you would find is if you had a cleaner that you introduced, even maybe one that's a little more gentle than hot caustic, if you're worried about environmental friendliness, if you were to take ATP swabs and compare your two processes, I think the story would be self-evident there.
Excellent. Thank you both. Luke, I was so pleased to see hose cleaning balls as part of your presentation. Not everybody in the Midwest has seen them or knows about hose balls. For anybody who doesn't know, you can order them through either PI Wine, Presque Isle Wine. I think the Vintner Vault in Paso Robles sells them. I don't think Scott Lab sells those cleaning balls.
Well, our sister company, Crush to Cellar, does.
There you go. Great plug there. But winemakers, you can get them in 1.5 inch, 2 inch, 3 inch sizes. And as Luke says, if you've never cleaned the inside of your hose, the first time you run one of these spongy scrub balls through your hose, you'll be amazed what comes out of it. And right here before harvest is an ideal time. I don't see any other questions. I'm going to make a couple other comments. Do either of you know the sort of the current cost of a handheld ATP reader and a pack of swabs? Last time I checked, it's about $650 for the handheld reader, and a pack of swabs was about $150, about $1 a piece.
Yeah. Go ahead, Luke.
I was going to say there's a lot of different ATP meters on the market, and they range just like a pH meter or a DO meter, any of the sort of stuff. Like you can get something that is, probably not very good at the job, but does part of the job. Whereas, I prefer things that really give me some precision, and then those are going to cost a bit more money. So I usually tell people it's around $2,500.
Okay.
Cory.
Yeah, we used a couple different ones throughout the tests. I would sort of echo what Luke said. You could get the job done for really less than 500. There are some entry-level ones out there. You'll sacrifice the sensitivity a little bit. I didn't see in our tests that, one would say the surface had nothing on it and the other would have a big boom and peak. Generally, they are in line, but you do gain a lot of sensitivity, when you do upgrade. The one thing I will say, Drew, is I'm looking at the Q&A here, and I actually do see maybe 4 or 5 questions. Is everyone else?
I have
okay.
more questions, but maybe,
Okay.
Drew, you don't see the Q&A. So I'm going to
Yeah, I can see it too.
So I will start by what was the brand of the white scrub pads and also the brand of the color-coded scrub brushes?
So the white scrub pads, the most common manufacturer would be 3M, but like if you go to Crush to Cellar's website and just put in white scrub pads, they have them. I don't know if it's an off-brand or not, but I wouldn't get bogged down the weeds on whether it's got a Chevy emblem or a Ford emblem on that. And then the color-coded scrub brushes, they look like Union Jack to me, but Cory, you might want to mention.
Yeah, those were Perfex brushes. Perfex is the name of the company, those particular model, but like you're saying, don't get bogged down in who the brand is. Focus more on how they're constructed, and really it's the injection molding part of it that's critical, and then the color coding just to separate, separate your streams like you would do for a bucket if you're doing big batches of cleaner versus sanitizer versus water.
Mm-hmm. Yeah, Drew, I'm unmuted.
Sorry, the other company is Remco. R like Richard, EMCO, makes all those sanitary brushes, shovels, rakes, any plastic, food grade plastic tool you use in winemaking. Remco is a great company as well. There's a person asking about sanitizing bungholes and barrels. I wanted to talk more about barrels. Cory does a lot of work with barrels. If that person, Bradley, would email me, I'll send you a presentation and answer some questions personally about how to clean barrels. You guys got to mention your pet peeves. I'm going to mention my Midwest pet peeve, and that is floating lid tanks. If I have to go to another winery and see a stack of floating lids that still have all their gaskets attached, all their vents attached, and a lovely cloud of fruit flies flying around them, I'll scream. So anybody who's using those floating lids, please take them apart after you use them, clean each component separately, put them away separately. Only when you get ready to use one of those floating lids, put the gasket back on, put your vents on, clean it and sanitize it before you let it touch your wine. And if anybody has questions about pump overhauling and how to properly clean your pump, you can reach out to me personally as well. I've got some practical experience in that.
So I will just keep going on the question we have in the Q&A, Drew. So we have one for you, Luke. Luke talked about using 10% water per tank size. Is this applicable when doing a circulation cleaning from the bottom valve to gamma jet? For instance, 30 gallons of water and 1 gallon of water gets the concentration 3%. If pressure is good, is that enough?
Yes. So really, it's 10% of the volume of the container when you're talking about automated systems like that. But what's really going to be the take home there is making sure you have enough surge volume of cleaning solution to feed the pump and not cavitate it while you're running it. And that becomes a bit of ongoing maintenance while the cleaning cycle is taking place, because the way I like to personally set up a cleaning cycle is all the valves, all of the ports, everything are open to an extent, and even like butterfly valves and ball valves, like at a 45 to clean the inside to at least get some action on it. And so you will eventually lose some of that cleaning solution. So maybe partway through the cycle, you'll have to re-up there. So yeah, that's what I'd say about that. But there's a couple other ones I see here. D-Stain-X question mark, sanitizer question mark. As far as back pressure, can you create a loop off of a pump and add a butterfly valve to create the necessary back pressure? Back pressure on a valve, correct? Yes, you can. I like to make sure that the end of my hose run, or however I'm circulating, is the high point in the system, so that you get all the air out of the system. That has to do with entrapment zones and surface area. Sanitizers, the industry needs to start looking at FDA recognized sanitizers, right? That's where we're going. PAA being one of them. There's some great information about PAA. It's been used in the brewing industry for many years. D-Stain-X is not an FDA approved sanitizer. Does it have some activity towards that end? Yes, it does. But it kind of exists as a cleaner and a sanitizer, and it kind of has a cult fan club around it in the industry. Once you start using it, like people start taking it home and using it in their bathtubs and in their kitchens and all sorts of stuff. So that's all I'll say about that.
Yep.
Steven,
any recommendation for the ozone generators for a small winery? Biggest tank is 1,000 liters.
No, I mean, the ozone generators that I'm aware of are larger size. McLean Ozone in Napa is kind of like one of the most common. They may have a small unit. So that's about all I can say about
that.
Can you hear me? Yep. Carlson also has a smaller ozone generator. And of course, they have such great customer service. Luke, you mentioned coated versus uncoated sodium percarbonate. Would you repeat what's appropriate, what's not?
Yeah, and the easiest way to tell is when you have a percarb and it's really slimy when it's in solution, it's really hard to rinse off. That's not the one you want to use. You want something that will actually have, like, it doesn't want to stick onto surfaces, right? And that's where, like, our D-Stain-X product, which, I mean, I'm not trying to corner the market on percarb here. I'm just saying that D-Stain-X has all the things in it that allows it to rinse really cleanly, which, when you get back to the efficiency argument, allows you to use less water. Now, in the Midwest, maybe that's not such a huge concern, but it should be globally, and it definitely is on the West Coast where Cory's at. Just incidentally, one of the reasons why I left California. They don't have any water.
Yep. So regarding the back pressure, what do you do for back pressure to make sure your hose fills completely with water when using a pig? Just elevated hose?
So yeah, the end of the run being elevated is probably the easiest way to do it. But also, when you run a pig, you'll naturally create the back pressure, and you'll fill up the hose.
And a last question: what is a good cleaner to use on concrete?
I will say Dystanex, but when I consider the cost of it compared to like how much square feet of concrete you probably have, probably want to go with just a straight sodium percarbonate peroxyhydrate available in 50-pound bags. And what I like to do is just make up a solution and throw it in like a little backpack sprayer or a garden sprayer and just rinse everything down and let it sit there, scrub it, and wash it off.
I'll agree with that. Percarbonate does a great job cleaning most concrete.
Okay. I have one question for you, Cory. When cleaning a barrel, so how long do you use the steam?
Yeah, so there's been some good research out there from Washington State. I think the author is Cartwright, but it's Dr. Edwards out of Washington State, for those who are familiar with folks doing research out there. He did probably the most robust study so far, and this was in the context of inactivating Brettanomyces at depth. So basically what they found was it would take 12 to 15 minutes of steam, basically boiling temperatures, almost boiling temperatures, to actually get the barrel wood temperature to reach 50 C, the inactivation temperature for Brettanomyces, for the right amount of time. So that's getting into it a little bit, but the right number of D values to reduce the risk. So 12 to 15 minutes was the answer that they found, but it really depends on what your goal is, because once you detect Brettanomyces in a barrel, unless you have really large format oak, like a Boti or something like that, that you really financially need to recover, it's a tough pill to swallow, and you really take a lot of risk putting wine back into that barrel after it's had a spoilage issue. So really, I think you could probably get away with going below if you're just trying to clean your barrel for reuse and you got a nice healthy fermentation. But the answer that I can give just from the research standpoint would be it would really take 12 to 15 minutes to inactivate things at depth.
And I'll add to that, just like Cory's slide with the ATP testing and showing cleaners and sanitizers, steam is kind of that sanitizing step. You can't skip over the cleaning step, which is I really like high pressure hot water impingement on a real barrel cleaner. The barrel cleaners that go inside and up into the bung and just kind of spin and it kind of just coats the surface with water. That's not cleaning. That's just wetting. So you want something that is high pressure and really creates that. Think of it this way: when you go wash something off, like your car, it's got mud on it, you take a hose, do you soak it, or do you put a nozzle on your hose and make a really strong stream? And same thing goes for inside of a barrel.
Yeah.
I'll even take that one step further and say that not only is that step important, but steam can be incorporated to actually help that cleaning. So I know at facilities that I've worked at, we would go through, just like Lou said, with a really, really high pressure jet. But then we'd actually put steam wands into the barrels for 5 minutes each, close them up and allow that vacuum to form. And that really effectively helps reduce or pull tartrates from the barrel surface. And so then you actually go back in with that high pressure and rinse out the barrel one more time. And then from there, we just actually used sulfur from that point because the barrel was clean, and the idea of then sanitizing it thereafter, if it was, again, a healthy fermentation, didn't make much sense. It just seemed like a waste of time. So in that context, steam was incorporated into the cleaning cycle, but as a loosening agent, really.
Okay. So I think we will stop now, just because we are like 11 minutes over time. Thank you to both of you for presenting. You did a great job. Hopefully everyone like it and learn a lot of things on way to clean, sanitize, and sterilize everything in the winery. So stay tuned for the next webinar that will be in 2 weeks. So August 18 at 3 p.m. Central Time. So same time, that will be on the management of sulfur dioxide with Dr. Gavin Sachs from Cornell University and Katie Cook from Scott Laboratories. If you have any question that we did not answer, feel free to reach out to us. So either to myself at watrelot@iostate.edu or Drew at dhorton@umn.edu. Or if you have the contact information from Luke and Cory, feel free to reach out to them. And so thank you all for participating, and we look forward to seeing you again next time. Thank you all. Bye.